they are very close.
Welcome everyone. Thank you for coming. So we will be starting. So welcome to this new webinar of the Ecology and Evolution in Montpellier. And today I have the pleasure to host Antonio, and I will let Sylvie introduce Antonio. Thank you very much. One thing, if you have questions online, please use the Q&A tabs, and we will switch on your mic. Thank you.
Okay, thank you very much. So, hello everybody. Well, I'm very happy to present Antonio Vazquez for this webinar. So, Antonio is currently researcher at Vazquez lab, where he was appointed more or less a year ago. And previously, he was head of the Ecology lab at the Pedro Guri Institute in Havana. And he specialized in epidemiology of trematodosis and other helminth diseases. He combines approaches in ecology and population biology. He focuses mainly on the interactions between mollusks and helminths. He also studies the diversity and ecology of mollusks of medical and veterinary interest. So it's your moment. Enjoy.
So thank you very much for the organizing committee of the seminar for letting me present a piece of what we have been doing and we are currently doing on mental malacology. And thank you very much to Steve for your kind presentation. Just kind of like to go to the field and something. So today, what I want to present you is something about several work that we have been doing related to the transmission of one trematode species that have a lot of intermediate host names. So we'll be talking about host-parasite interactions, and I believe that you will know that between host and parasites, usually there is a strong or a tight ecological relationship that's established given the usually strong evolutionary history between these two partners. So this will ultimately define the eventual susceptibility and compatibility of each system, of each parasite and its host. And these systems occur across virtually all living organisms. But today we will be focusing on snails and trematodes. So speaking about specificity in host-parasite interaction, and what concerns trematodes, they have tightly evolved with mollusks, and there's only a handful of cases in which a trematode is using an annelid species as an intermediary host. So if you see in the phylogeny of Trematoda within the DNA, they practically use only mollusks, and within mollusks is the class Gastropoda, which is snails. So I believe that snails, trematodes will need more fine introduction. We're talking about one of the classes of Mollusca, snails, and in the case of trematodes, they are the flatworms belonging to the Trematoda within the phylum Platyhelminthes. And what is particularly interesting in this in the interaction with these two entities is that in the very, very diverse life cycles that exist across the Trematoda species, you can see that you can have usually an intermediate host, then you have final host can be a mammal, a bird, amphibian, also another invertebrate. You can have some states that will live outside the host for a small amount of time, so larvae that will seek actively their intermediate final host. But even in the most reduced life cycle of trematode, there is always so. Sometimes everything goes on in the scene. And of course, both snail and trematode system are great model for evolutionary studies given this strong interaction between them. But they are also very important, very interesting because they are what we call, or they provoke what we call the snail-borne disease. And I believe that maybe some of you have already heard about some of these diseases, like schistosomiasis with the blood flux, like paragangliosis with the lung flux, or like all the varieties of hepatitis B stomatosis with the liver flux. And it's precisely of these last examples that we will be talking today. So we will be talking about fasciolosis, we will be talking about a trematode that is called Fasciola hepatica. And fasciolosis is a disease that provokes, that exists everywhere in the world. It's the largest distributed trematode with the highest altitudinal, longitudinal, and latitudinal distribution. It affects today, and all these numbers that are highly underestimated, affect today 35 million people everywhere, with over 500 million of domestic mammals infected, and it costs over $3 billion every year for the economy of the countries where the disease is present. And it's not only the evolutionary relationship that shapes the outcome of fasciolosis, but it also has strong socioeconomic relationship that dictates that in the world there are several human hyperendemic areas of fasciolosis. For example, the one that occurs in the Bolivian altiplano, where there are localities with over 40% of the population that is infected with fasciola, and half of the population that is infected with fasciola at school age. So it's really a big deal as it is. So I will start by presenting you the partners in this snail trematode interaction. So I will start with the parasite. This is Fasciola hepatica. I don't know if many of you have seen this parasite before. Here you have a picture of the dissection we made in Cuba of an infected liver cow, where you can have the parasite coming out of the liver. I think I got something for you that you can pass on. You have never seen Fasciola. Here you have Fasciola hepatica. In adult, you don't have to open this. And so, as I said, it's a leaf-like parasite of around 2 to 3 centimeters long. And in the evolution of fasciolate, and this is important, early fasciolate evolved using strictly aquatic freshwater snails of the family Planorbis. But actually, even the most vast species that occur today, like Fasciola exclusi and Fasciola reruta, still use Planorbis snails as intermediate host. But eventually, there was a host switch from Planorbis to Limnaeidae. And Limnaeidae is a family of freshwater snails that has fully aquatic species and species that range from semi-aquatic to very amphibious species. And this facilitated actually the fact that Fasciola started to use other final hosts that makes that today the species of the only 3 species of Fasciola that occurs today, which are Fasciola niansi, which only infects hippopotamus in Africa, Fasciola gigantica, and Cosmopolita Fasciola hepatica, which is the topic of today, only use leaf-laying fruits. I will not walk you through the whole life cycle of Fasciola hepatica, so briefly, the adult will live in the biliary channels of the liver of mammals, where they will be an hermaphroditic parasite. They will reproduce sexually. They will produce thousands of eggs on a daily basis. The eggs will leave the final host with the feces, get to the environment. They will hatch, conditions are favorable, and a ciliated larva that's called metaphyll will exit the eggs and will actively search for a snail. Again, and I will repeat this every time, eliminate. So once the larva enters the snail, it will transfer into sporocytes, then into relia, and thanks to a tremendous sexual amplification, it will produce several generations of the relia, and then finally will produce a tail larva that's called Cercaria, ex snail, and will insist in the environment in the form of metacercaria, waiting for a final host to enter the seed and get infected. So it will exist in the duodenum, migrate to the liver, maturate, and so on. And this is the cycle of Fasciola hepatica. So, for the final host, I won't say much because we will concentrate on the intermediate host, but after review that Anya did a few years ago, if I am not wrong, I think that what we know today, Fasciola actually is able to infect 45 species of mammals that we know naturally across 7 orders within mammalia. But it's true that most final host Fasciola are within the actual. But it also infects plants, it also infects us, as I said, and the earliest record I found for fasciole hepatitis infection of humans date back to around 8,000 years before BC in our geoparasitological finding site. So, fasciola may live in the liver of mammals for several years, it's protected in the liver. So the window for transmission toward the intermediate host is fairly open because an infected mammal will always produce eggs that will go out to the environment. However, this is not the same for the female. And this is why we always say that snails play a key part in transmission ecology of the parasite. Because for the parasite to be transmitted from the snail again to the final host, it will depend on the species of the snail, and compatibility of a particular species in the numbers, the effective number of the population of the snail. So it will increase the probability of transmission from the snail to the parasite. So we get to the snail, and as I said, all intermediate hosts of Ceratidae are within the Limnaeidae. This is a very interesting family. We have a huge diversity, and it's very diverse in everything. There are species that are widespread across the world. There are species that are localized in a smaller areas. There are species, as I said, that are fully aquatic. Here you have, this is a picture that we have taken here in France, or in Southern Europe. You have Ampullaciana baltica, which is strictly aquatic. Then you have Orientogalma vigidis, which is in Spain, which is a species that is semi-aquatic. And then you have the quintal transmission in Europe, which is Galba truncatula, which is very, very, very amphibious. You have species that prefer to reproduce by self-fecundation. You have species that prefer to reproduce by outcrossing, some that will live for a few months, some that can live for several years. So if you see this phylogeny of the Limnaeidae, you will see that they are hosts of either Fasciola gigantica or Fasciola hepatica, mainly Fasciola hepatica, all the way across the phylogeny of the Limnaeidae. But in this phylogeny, I will concentrate on a group of snails, which is the Galba snails. Why the Galba snail? Because these group of snails have demonstrated that one of the most important on the epidemiology of fasciolosis. As you see, and this diagram here shows what we have recorded in terms of experimental prevalence in the lab using a lot of Limnaeidae species. So please concentrate in this one here because the other is Fasciola gigantica. So you see that the species that show the highest prevalence record are the Galba. So who are these Galba? These are tiny. I have something for this also. So these are really tiny muddy because they like the mud, but especially cryptic species, and this complicates for us to study the epidemiology of fasciolosis because it's very difficult to reliably differentiate between one species of Galba to the other. There's a lot of variability across the 10 species. Thankfully, we have a colleague in our team, that developed a molecular toolkit using a differential amplification of microsatellite that allows to discriminate at least among 3 of the most complicated species. Why the 3 of the most complicated species? Because we can be sure who is Galba cubensis, which is the main intermediate host in the Americas. Okay, we have Galba truncatula, which is the main player for fasciolosis in Europe, some places in Africa, but also the main responsible for the human hyperendemic areas in South America. And then we have Galba shiracensis, which is a species that possesses a species that tends to disengage the transmission of fasciolosis. I will be talking later about this. But the thing is that Galba snails show their highest diversity in the Americas, and most of the species we are sure they are native from the Americas. That's also because Fasciola is supposed to have originated in Asia. So we have told ourselves, how is it possible that one of the main intermediate hosts are from the America, but Fasciola is from Asia? And the story we had until recently was that Fasciola goes out of Africa with they mass in Asia and will eventually enter the America. But the story that they have tell us was that it came with European colonization 500 years ago. And during this trip, the idea was that Fasciola alum, it came in the liver of cow, and we're okay with that. But then they also say that the main intermediate host that we have always supposed to live is Gabas truncatula, which is largely distributed in Europe, came attached to the feet of sheep and cattle. So that we have to imagine that Gabas truncatula had to endure a 3 to 4 month trip in a 15th or 16th century old ship. It's complicated, but that's the story we have. Until recently, there are molecular evidence that shows that Fasciola hepatica existed in South America, in Patagonia, actually, 2,300 years before Christ. So way before the arrival of European colonization. And when we saw that, we said, okay, so maybe there is an alternative hypothesis. So the first image that came to our mind was this. Because we knew that the cousin species, which is Fascioloides magna, is native to North America. So we said, okay, if we have Fascioloides magna in North America, maybe Fasciola hepatica came in the same way the ancestor of Fascioloides came to the America. So what we proposed, briefly, we'll try to be sure on this, is that yes, actually Fascioloides emerged in Africa using planarby snails. They infected proboscidean. It was, as I said, the moment in which there was a hot switch toward living nail. They would go out of Africa with the early migration of proboscidean into Asia. It will diversify there thanks to the diversification of mammals. They will get reintroduced in several parts of Europe during mammal migration, and they will eventually get into the America using the Bering Land Bridge and find in the American limnaeus snail until they will colonize all the American continent. Of course, the epidemiology of fasciolosis has been largely increased in the last 10,000 years since the Neolithic with the domestication of mammals. But it will explain its much more early presence in the Americas. So when Fasciola hepatica gets to the America, what do they find? Well, we find a lot of limnaeus species, but mainly two types of species. We'll find the Gauta snail that I just presented you before, and you will also find the North American Celosuccinia cormela, which is a semi-aquatic limnaeus species that today plays a very, very, very important role in the transmission of fasciolosis in this native area and everywhere in the world, because they have been introduced virtually everywhere. I think only except in Asia, we haven't found any record of cases. So, to what concern the Galba snails, here I show you some natural prevalence. This is natural prevalence that we have recorded in the Americas. And you see that there are many species of Galba that are able to get infected with Fasciola hepatica. And maybe when you see this number of prevalence, you might jump to the idea that they are very low. But actually, what we have shown in several studies that we have carried out in Cuba is that even if you have very few snails that are infected with Fasciola, these very few snails can harbor around 50 to 120 radiates, and each one, as I said, can produce between 8 to 15 cercoria. So they suffice to maintain an infection in cattle that range in Cuba with the same numbers between 70 to 100% infected surveillance. So then there was the puzzle of the ecology of Galba snails, because what we have always, what we have learned about fasciolosis, studied life cycle in the school, is that this is an aquatic life cycle, that it will only happen in the water. But the thing is that when we go to the field to sample Galba snails, this is what we find. This is what we found. So what you see here, this bunch of people here in all four, which is actually the classic molecular position, and which is, by the way, us in the field, is people that are looking for galba snails, the top of the hill, 300 meters from the nearest water source. And that's not what they saw when we learn Fasciola capital life cycle. And here you can see an IQ sample snail again in the mud. Yeah, there is humid condition, of course. We're not talking about a land snail, still a freshwater snail, but only humid conditions suffice for the snail to get infected, develop the parasite, and leave the cyst in the environment. And this, of course, will change our idea of the transmission of Fasciola, and will enlarge the epidemiological risk of Fasciola transmission. So we have the galba snails, which are made in the Americas, Celsusinia columella, which is also in the Americas. And now we have a problem because in the last couple of hundreds of years, we have seen a massive introduction of everything, but for what matters today, snails. So we have galba truncatula in Europe since probably very long time. It's the main player of Fasciolosis here. It's a species that is adapted to temperate conditions. So it's a species that will have population will depend on the temperature. And now we're having several species of Linnaeus snails that are coming. So there are two galba already in Europe. We have Galba guensis, which is present in Spain. It was previously only recorded in the delta of the Ebro River in Spain. Now, 1 year ago, we sampled in the outskirts of the Barcelona airport in Llobregat, and there is a recent notification of Galba guensis in Azerbaijan have been published. But also we have Galba shiracensis. Every time you go to the field now here in France, we find Galba shiracensis. So it seems that it was more spread than we thought before. And we also have Celsusinia columella, which we knew was in France since more or less 20 years ago, but we didn't know that it had spread so much. And we are now sent to a project that is led by Anya. Next to some project, we are following the invasion of Celsusinia columella in France. So we have more species here that are very good hosts for Fasciola hepatica. And these species like to travel or can travel very well attached to the plants, to the aquarium plants. So the plant, the aquarium plant and petrate can play a big role in here. I did a quick search in the internet, and actually you don't have to go to a pet store to buy a snail. You can buy it from your home through Amazon. There are prices for everything from some Linnea. You can buy it in Amazon, you can buy it in eBay, in AliExpress, everywhere. So there's a big traffic of snails, and with the snail, there's danger that comes associated to that. So if we talk quickly about Celsusinia columella, we see that it's been using the hydrographic network of southern France to invade this region, mostly associated with Canal du Midi here in France. Here you see columella down in Loire River. Here is in the concrete walls of Canal du Midi. So we are on to that. And what concerns galba snail, well, as I said, Galba guensis for now seems to be restricted to Spain. There's this prevalent in Azerbaijan, but we haven't found it yet here in France. But to what concerns Galba shiracensis is very interesting because, as I said, we find it not every time, but more or less on a regular basis to find in the field. And the species is always close to us because, as I said, we have been having for several years now this hot discussion on if it is an actual host of Fasciola or not. So there's several studies that have been carried out to try to decipher this question. And there are people who doesn't manage to infect the snail and say it's no host. There are teams here in France that managed to infect it a little bit and say it's a potential host. Me personally, with our team, we have been trying to infect this species like for now 15 years ago, and we have never succeeded in infecting this snail. But we have always said it might be a host if conditions are proper, because this has actually been found infected in the field in Ecuador. So the problem is that the epidemiological implication of a no-host classification would mean that it is safe if the species present is scholarship. We were not okay with that because it's dangerous to say that. So we tried to challenge this hypothesis. And because we were failing every time we tried to reflect Ecologie-Evolution with Fasciola, we said, okay, let's try something else, because the argument, the people that say that it's not a host, is because Ecologie-Evolution is the garbage species that display the most amphibious behavior. So they base their argument on the fact that because it is the most amphibious, practically land snail, there is no common evolutionary history between Ecologie-Evolution and Fasciola hepatica to make Ecologie-Evolution compatible with Fasciola hepatica. And we said, okay, but let's try something else. So we need to find another trematode species that has the same transmission ecology as Fasciola hepatica, and that uses lignacin. And we had it here in France, which is the rumen flu, Calicoforum dominei, which has exactly the same life cycle as Fasciola, except that it goes to the rumen and not to the liver. That is the same snails, it's the same final text. So we did it, and surprise, it get infected. So this story of it cannot get infected with a parasite that transmits in the water doesn't exist anymore. And when we saw this, we said, okay, so it get infected with Calicoforum dominei, rumen flu. Let's see what happens if we perform dual exposure between Fasciola and Calicoforum dominei. And long story short, we tested several different ways of infection. So we exposed them at the same time. We exposed them first with Fasciola, and then with Calicoforum, and then we exposed them first with Calicoforum, and then with Fasciola. And we found that it can get infected with Fasciola if it is first infected with another trematode, in this case, the rumen flu. And here you can see this is a radio Fasciola hepatica in Ecologie-Evolution. These are Cercaria. These are, for example, metacercaria of Calicoforum dominei. Now, prevalence were not very high, and this was the first study. We need to repeat this. We need to share the conditions. We need to optimize our infection trial. But it says that Ecologie-Evolution can actually get infected with Fasciola. So this justifies the natural prevalence that have been found in Ecuador. So maybe there is a mechanism of facilitation if this species is getting infected with a compatible trematode before Fasciola finds the field. So I want to finish with three hypotheses that we have now. We will be working on this in the near future. This is just a schematic. So in green, you have, let's call it the local species, Calva truncatula. We know very well the ecology of Calva truncatula. We know this species that peaks twice a year, mainly in spring and then up in autumn. It's a species that likes to live more or less in the land-water interface. But in the case, Vazquez and Ecologie-Evolution are actually equations. Now we will have the probability of transmission will increase even in summer because we are talking about tropical species. In the case of Pseudotsuccinia columella, which is much more aquatic than Calva truncatula, it will increase the probability of Miracidium snail encounter in the water environment. And if parasite facilitation plays a role in the case of Calva chilacensis becomes a good host of Fasciola in Europe, it will expand the risk of transmission from water to more land environments. So we foresee with all global change, particularly climate change, that Fasciola might tend to increase in Europe, given the arrival of these tropical snails introduction. So just to tell you that you have to be careful with what you eat. We'll finish with that. I thank you very much. I want to thank particularly the team that have been working with him for almost 15 years, and also the lay students that have been working especially with GABA and sustainability. Thank you very much. So you have questions. Any questions? Yeah.
Thanks for the really nice talk. You have an idea of how this priority effect can work, or one to facilitate the other?
What our guess is that once the parasite gets into the snail will tend to fight back the parasite. So the immune system of the snail will say saturated with one parasite and will less start the other. We have seen, we saw that a few months ago. We really need to start learning what is the mechanism of, if there is facilitation, what is exactly how it works facilitation. That's just a hypothesis that we have.
Any other questions in the room?
I relate to the first question. Do you consider estimating abiotic stress, like temperature, pH, on the resistance or the immune response of the snail?
Yeah,
That would be
yeah,
yeah,
as we said with another species, with the Ecologie-Evolution, we have challenges with many stress and different stuff, but we have a really tough population of this species in Cuba. Yes, we plan to play with this species a bit, see what happens, see if it's only because there is a parasite, or if any other stressor might also give the same outcome and improve the susceptibility of this species.
I think that the solution might be facing much more terrestrial habitat than we produce. When we test this rate, and then we usually compare snails.
Yeah,
and hosts in the water. That's
yeah, thank you very much. That's something that I didn't want to put here number because we haven't published it. But yes, we carry out what we call at the time amphibious inflation. So basically, we prepare mud in a petri dish. We put the eggs to hatch. It was not dry mud; it was humid mud, of course. But remember, it is an aquatic parasite. So we let the eggs hatch. We put the snails. We let the snail pond. So we proved that Miraflor was okay, but Miraflor was not able to swim because there was for no water. It was the snail that came to Miraflor and was the empathic bacterial attached. And we did the same thing in our current infection trial, so in water. And we found no difference, no significant difference in prevalence or intensity. So we proved that happens. Which is more complicated, for the part in which the snail will shed in land environments. We haven't tested yet for fasciola, but we just tested with a team of colleagues with the University of Munich that did it with Gallagher tests and the rumen flu, which has the same life cycle, same transmission ecology. And they proved that the Gallagher sensitive, in fact, the Gallagher sensitive with the rumen flu will tend to go up, and will give you like 150 meter area in 2 square centimeters, and it will shed in the air, not in the water. So if you can imagine if they do this in the grass, then you see how carp will come. Perhaps that will justify we support the fact that sometimes when we go to slaughterhouse, we find cows infected with over 300 bacteria. But imagine if you, in a single event, you ingest 150 meters of period. That match with the number that we find.
So it depends for the size. Or in this environment. Ah. Well, this
House test. We don't know. Problem.
Can be better.
Yeah, maybe it's just plasticity, maybe there are some kind of selection, I don't know. We could test it, for example, in region where transmission do always get using a fully aquatic snail, like in Africa with Stratus naturalensis. We could test there, but maybe could be interesting to test it just plasticity or if there is some kind of selection toward this more active.
Other questions in the room? Online, perhaps? So just to follow up this question, so what are you expecting in terms of adaptation? Is it like losing their swimming ability, for example, or like the Miracidium or the Cercaria?
I won't say that. I would go more for plasticity because if this is true, and these are hypotheses that we can need to get it in the lab, it has been happening for a long time. And they still circulate that they are still get serious. So we need to toward plastic or down.
If I can give an idea on that, I would guess that it could go through a shorter time of enhancement. So the larva that can go through the cysts will develop more rapidly than in the one that is more active.
What we have observed is that in the experimental infection of the mesophaseolus for several years now, actually, when it is not shed, it will exist less than a minute, sometimes seconds. You cannot imagine a fasciolated parent that will swim and be fine, but it lost the most. No, it will. You put your fingerprints. So it's, that's what we have seen.
Other questions? You talked about Africa. So what's the situation in terms of introductions or invasions of those three tropical species coming from Latin America, South
Well,
America?
we don't say that the one we have here, it came from Latin America. So, for example, Susina columella. No, they are native from there.
Yeah.
But they have been introduced everywhere. So maybe Susina columella that came from, I don't know, Africa.
So they are actually introductions of those species.
Yeah, they are also introduced in Africa. Capatula is in Africa. The Susina columella was introduced into Egypt, where there has been a strong field back of Fasciolatus canicki, and it was recently found in Zimbabwe. And with a country, I don't remember the name. And actually, in Zimbabwe, is producing strong field back of Fasciola in the hippopotamus population. Okay. And I don't know if there is mortality rate associated with the introduction as well. But actually, what they say, they saw the introduction of the water hyacinth, the invasive toeco-epidemiological, which then were also producing a columella. And we have all population transmission. Faciola hepatica, faciola viva, and faciola mens, which is the faciola of people.
What's the effect of the on the cell of the environment?
Fasciolosis, as I said, would start in the liver, in the biliary ducts. It will give you anemia, it will give you inflammation of the liver, malfunction of the liver functions, classification of the biliary ducts. This is an extensive question because here several times I present this topic. I have questions even from doctors that ask me, but why is it important to study fasciolosis? Because I mean, it's true with a single dose of triclavendazole, which is the molecule of choice to treat Fasciola hepatica, you kill the parasite. It's 3 times that I have any question from the doctor. Yes, it's true that you kill the parasite, but the classification of its place, the eventual complication of having a tremor going to see the stage to tremor, and those are small. Fasciola can grow a little bit bigger. So you can imagine having 30 or 15 those worms in your liver vessel. And the thing is that in non-endemic countries like in France, if you get infected with Fasciola, in the acute phase of the disease, you will have like the general symptoms of any digestive problem. So you have abdominal pain, you can have some fever, you can have you can have some diarrhea, and this is the moment in which the parasite is migrating from the liver to the liver. So the doctor will treat you for a common abdominal disease or abdominal problem. The thing is that when you get to see the X in the physics, when you get confirmation of the parasite, there are not there. So that's why it's important to study, because what we want is to understand so we can prevent. So we don't have to go to the treatment of triclavendazole. And there is also the problem of resistance because of the overuse.
And in the snail?
And in the snails, well, in the snails, actually, it depends. It depends on the compatibility of the snail and the parasite, of the parasite strain, and strain. So if it is not compatible, compatibility is low, it can be very virulent, and can kill the snake. I think that is what happens with garbage testing. There is no good compatibility, so the parasite usually kills the snake. It will install usually in the gonads of the snail. So it will affect reproduction, though we haven't seen Fasciola to castrate the snail, but in any case, physically castrate the snail, but it will physiologically castrate the snail, it will give her generation to fight by the parasite, so it will not lay eggs, will not reproduce. So it will affect lifespan.
Yeah, thanks for any questions online? No. We have time for one or two questions. But okay, thank you.
Thank you very much.